This poster looks at movement-induced mechanical stimuli and how they determine joint shape in regenerating axolotl forelimbs. Previous studies have shown a necessity of muscles in order to form healthy skeletal structures. These studies show that movement-induced mechanical stimuli are critical for proper joint formation, but less is known regarding the mechanisms of how this actually works. In order to answer the question, what is the effect of elbow flexion on joint shape, we block chondrocyte mechanosensitivity through targeting the TRPV4 channel during limb generation in axolotls. A computational model informed by these axolotl experiments allowed us to explore how limb motion regulates joint shape and the many potential mechanical stimuli driving cartilage growth. Light sheet fluorescence microscopy images were taken of regrown axolotl forelimbs in order to quantify differences between a control group, shown on the left, and a group with blocked mechanosensitivity, shown on the right. Humeri and joint shape were analyzed along with the quantification of proliferative cells, shown in green. Cell counts were obtained using a deep learning algorithm called Stardust. Based on analysis of this experimental data, insensitivity to mechanics was shown to result in smaller condyles and fewer proliferative cells. Computational modeling was then used to identify what type of mechanical stimuli might affect joint shape and growth through a poroelastic model. Local tissue growth promoted by compressive fluid pressure due to loading was able to accurately predict the joint shapes in the axolotl experiments. In order to investigate how mechanics affect joint shaping and development, axolotl experiments were carried out highlighting differences between blocked mechanosensitivity and controlled joint formation through quantifying proliferative cell growth and shape analysis. The predictions of our computational model of tissue growth during joint morphogenesis suggest that interstitial pressure plays a role in regulating joint shape. These findings help us further understand the many mechanical stimuli that might affect joint development and morphology. Integrating experiments and computational modeling provides interesting insights that experiments alone can't deliver. In this study, we developed a promising framework for identifying potential biophysical stimuli through computational modeling for further study and experiments of how motion might affect joint development and morphology.